Hello friends, today me and my teammates are going to talk about application of biophysics in early drug discovery. Biophysical methods, which can encompass a wide range of techniques, focus on measuring the structure, properties, dynamics or functions of biomolecules, have been increasingly employed in the drug discovery process since their first introduction in the early 1990s. It was during this time that commercial instruments such as the first isothermal titration calorimeter and the first surface plasmon resonance instruments became available and their use exemplified using biochemical systems. Alongside this, existing biophysical approaches were used in novel ways to identify and characterize protein ligand interactions. For example, the first report of the use of affinity selection coupled to detection by mass spectrometry for the identification of molecules binding to macromolecules and the use of nuclear magnetic resonance to identify fragments that could subsequently be optimized and linked to form more potent compounds. This allowed the valuable combination of biophysics with HTS to contribute to the establishment of high quality, high throughput assays through the characterization of protein and tool ligands, as well as the evaluation of HTS output through orthogonal applications of biophysical methods to screen for true target engagement. More recently, biophysical methods have matured, throughput has increased, and sensitivity improved, such that some of these methods can also now be employed in primary screening not only for fragments, which typically screen lower numbers of low molecular weight compounds, but for screening libraries comprising many thousands of compounds. The high fidelity nature of many biophysical methods, coupled to the specific information content they can access, has also meant that they are used increasingly to characterize the mode of action of hits and leads. Some methods provide access to kinetic data, whilst others enable thermodynamic characterization of ligand binding. Others may allow structural insight into binding mode and binding site. asset development. The capability to design, build, and implement assets that are specific, robust, and sensitive enough to identify and characterize potential new drug molecules is fundamental to drug discovery. In developing a new assay, there are several factors that must be considered. These include the nature of reagents such as their identity, purity, concentration, functionality, and stability. The application of biophysical methods early during assay development can help to understand some of these features and to ensure that high fidelity fit for purpose assays and are developed. Of course, this extra resources needs to be considered during project planning, but the benefits of applying this early outweighs the resources that would need to be applied to rescue projects which have been misled or have failed due to spurious activity from poorly characterized reagents or assays.
Reagent quality control. Early application of biophysical methods often focus on understanding the quality of the reagents available, both in terms of the suitability of the target protein and the behavior of the non-tool compounds. For a protein to be deemed suitable to be used in subsequent drug discovery assays, it should fit criteria associated with the group of characteristics highlighted in the paragraph above. The purity of protein is also important, as impurities may have similar activity or bind test compounds. The concentration of the protein should be measured so that concentration dependent effect can be understood. The functionality of the protein should also be instigated. This may require measurement of ligand bonding, the catalytic activity, or the ability to carry out other function or interaction. Finally, the stability of protein should be assessed both in terms of its thermodynamic and kinetic stability, as well as its ability to withstand certain conditions such as freeze. It also may necessary to characterize the bonding of other molecules such as substance, cofactors, bonding partners, or competing probe molecules to understand their requirement and concentration range desired and permitted within the same methodologies under consideration. These attributes of the target protein may be investigated using a variety of approaches, and biophysical method can be provide confidence in target protein by being employed alongside or instead of standard biochemical method for protein. In this respect, it is useful to remember that the target protein goes into every well of the experiment, and so issues with the performance of this reagent has the potential to compromise the entire experiment. This can be contrasted with efforts to ensure individual compound purity and activity. Although individual compound integrity is important to ensure that the identification of useful chemical start points from any active hit compound is not missed, it does not have the same propensity to compromise the whole hit identification process, as would be the case for a protein questionable integrity. Protein integrity is and its resulting ability to bond the test compounds which may modulate the biological function can be compromised in several ways. The application of biophysics to characterize reagent quality may not be a large resource intensive effort. Often a single but decisive experiment can be extremely informative and often critical in understanding the behavior of the target protein. Additionally, since these methods are often relative, generic, requiring little assay development, their use at this early stage is not prohibitive and is often influential and impactful. Even if methods do require additional development time, for example, SPR assays may take a longer to establish than simple ITC experiment. This is usually time well invested, as the method are frequently used again for hit evolution, and so the development time is effect just pushed earlier in the workflow than it otherwise may have been. In the case of SPR, having an assay in place to characterize hits post STS is very valuable.
Hello everyone, myself Swati. Assay quality control. In addition to being able to characterize reagents, biophysical methods are also an invaluable tools applied to evaluating assay for their ability to identify compounds that engage with the target protein to bring about the declined effect. Any biochemical assay has the potential for artifacts to arise due to non-desired mechanisms. These may be specific mechanisms that are unwanted in a drug like compounds such as reactivity, redox cycling, colloidal aggregations, heavy metal contaminations, protein unfolding, protein denaturation, or biological systems or technology interference such as coupled enzymes, inhibitors, fluorescents, compounds, or quenchers. Understanding the liability of a biochemical assay to these types of compounds helps to understand the potential output from high throughput screens utilizing that assay approach. At AstraZeneca, HIT's development includes testing a small library of around 1,000 compounds with unwanted mechanisms of inhibition and around 7,000 compounds which are meant to represent the diversity present in the full screening set of around 2 million compounds. Biophysical methods such as SPR or NMR employed following these early screening activities to characterize the hits so that the knowledge rising from an understanding of the reasons behind false positives may be used to further optimize the assay or to avoid these types of hits in the full screen. Impact of biophysical evolutions of hits identified during hits developments. This workflow therefore allows decision to be made based on any specific issues that arise due to the natural this workflow therefore allows decisions to be made based on any specific issues that arise due to the nature of the assay that may be mitigated or avoided by modifying the screening cascade. For example, knowing that heavy metals, contaminants, or redox cycling compounds may be hits in the biochemical assay, but the compounds are not true binders to the target proteins in a high fidelity biophysical methods allows the primary screen to be modified to reduce the liability to such effects. Primary screening. There are few biophysical methods that can be applied to primary screening as usual defined in the high throughput setting, which often refers to the testing of 1 million or more compounds, and usually these methods are more frequently applied for secondary hit evaluation. The predominant reasons are the amount of protein required and the throughput required to achieve primary screening in a reasonable time frame. To address these issues, compounds mixtures are often used to facilitate reductions in reagents and time, since many compounds may be screening from a single well. Often, mixtures have no adverse effects on the protein or on the ability to detect binding, but sometimes problems are experienced if there are compound-compound interactions or if the compound organic lead as the compound solvent concentration is too high. Two biophysical methods that are often capable of providing throughputs approaches that of traditional hits are affinity selection, mass spectrometry, and thermal shift assays. These methods utilize mass detection of ligands bound to a target protein and the ligand induced increases in thermal stability respectively to identify hits. The benefits of these approaches in primary screening is that the assay is generic and can rapidly be optimized for the target of interest. The disadvantages in biophysical primary screening is that identified hits may bind to the target protein but may not have the desired biological effect. An alternative use of biophysical methods in primary screening is in testing a much smaller library of low molecular weight compounds in so-called fragment-based screening. This was first described employing NMR screening to identify fragments that could be subsequently be linked and optimized to make higher affinity compounds. More recently, fragment screening has been desired by other biophysical methods in including both SPR and TSA.
High throughput screening, affinity selection mass spectrometry. The application of mass spectrometry in the drug discovery process has been well established for many years. It has been applied both in the characterization of market proteins, where it has been primarily focused on quality control, as well as target identification and validation. For small molecules, the technique has been used for metabolism and pharmacokinetic studies as compound identification. More recently, MS approaches have been developed to detect and characteristics protein small molecule complexes. Affinity selection mass spectrometry is now the predominant biophysical method used for primary screening. It was first introduced in the late 1990s and various formats have been described, evaluated and implemented since then. However, the basic premise of all these methods is the detection of compounds that bind to the target protein using mass detection of the bound ligand. This simple binding assay has been benefit to reduce interference such there is no requirement for functional activities which often entails more complex assays and therefore often increased probability of effects arising due to effect on the readout rather than from true target engagement. Another advantage is that this screening approach can be applied to orphan and unique targets and target for which functional assay can be developed. The major disadvantage alongside the requirement mentioned above for assigning functional activity post-screen is that these methods are not commercially available and systems enterprise and sometimes even the software solution required for data analysis have to be developed in-house. Although there are suitable differences in the way as MS methods may be employed, the principle of the different approaches is essentially the same. High throughput screening, thermal shift assay. The thermal shift assay, also known as thermofollicle or differential screening fluometry, has been used for several years to study protein stability. It is a rapid and simple method that allows the melting temperature 50% of the protein to be determined under different conditions. It has extensively been used to optimize buffer condition for X-ray catalogs studies on soluble proteins such that those conditions yielding the highest stability may be used as start points for crystallization trails. The addition of compounds that bind to the target protein thermodynamically stabilize the protein relative to control in the absence of lesion, and this stabilization can be used to identify binders from non-binders. The TSA experiment involved incubating the target protein with test compounds in the presence of a dye that binds to hydrophobic regions of the protein. The temperature is then increased uniformly and the fluorescence of dye monitored with temperature. As the protein unfolds, more hydrophobic regions are exposed. There is increased dye binding and the fluorescence intensity increases. Compounds that increase the thermal stability of the protein may be identified as those giving an increased TM, resulting from a shift in the unfolding curve to higher temperatures. The advantages of TSA as a primary screening method includes the simplicity of the approach, the cost effectively and the potential to access a wide range of binding affinity. The method requires little assay development, which is in effect limited to adjusting and to give a suitable signal. The reagent requirements are protein and dye only, meaning that the costs associated with expensive biochemical reagents are avoided. The method does not require specialized instrument and it carried out using standard thermocyclers, often used for real-time polymerization chain reactions.
Fragment-based screening, fragment-based drug discovery is now a well-established approach with FBDD having delivered two marketed medicines, vemurafenib and venetoclax, and around 35 compounds currently in clinical trials. Identifying fragment hits is a challenge in traditional biochemical assays since they are likely to bind weakly to the target protein and the apparent affinity may be weaker still due to competition with substrates. Biophysical methods are well suited to fragment-based screening as they are sufficiently sensitive to detect weak interactions and reliable enough to avoid false hits. Historically, techniques including NMR, SPR and X-ray crystallography were applied to fragment screening, but more recently methods such as microscale thermophoresis and TSA have also been used. Compared to traditional aids, the throughput of fragment screening is generally lower, and the biophysical methods employed often require larger quantities of protein. Fortunately, this issue is overcome by the ability to screen fewer molecules, but to cover much larger proportion of chemical space using fragment screening libraries. For example, screening 1 million compounds out of the estimated 1030 compounds that could potentially be synthesized with 36 heavy atoms, around 500 Da, is 10-19-fold less efficient than screening 1000 compounds from the 108 potential compounds with 12 heavy atoms, around 160 Da. Another way of considering this is to reframe the coverage of chemical space by considering how many compounds need to be screened in order to obtain a sufficient number of hits against the target of interest. Thus, the probability of detecting binding for ligands of different sizes is expected to be low for very small ligands due to detection sensitivity, high for small ligands, and decreases with size for larger ligands due to increasing probability of steric clashes between the ligand and protein. This suggests that focusing on fragments with lower heavy atom counts with the most sensitive biophysical screening methods provides the highest probability of allows a small number of representative compounds from each cluster to be tested in a suitable biophysical technique. Positioning biophysical testing early in this way allows focus on those compounds that are confirmed by biophysical method and deprioritizing those that may likely be false positives. In this context, biophysical methods provide a means of selecting compounds based on positive selection criteria such as target engagement, mode of action, and for some methods, even functional activity. MS, NMR, and event ITC can, for instance, be used to monitor inhibition of substrate conversion for enzymatic reactions rather than the negative criteria associated with filtering compounds for unwanted mechanism of action. An important consideration is how biophysics might be used in the confirmation of its resulting from cell-based screening. This is different for the case of cell screens, where the biophysical approach may be considered less physiologically relevant than the cell assay. Biophysical methods still have value in confirming binding and providing additional information, but extra caution needs to be applied in comparing the results and in making decisions about fate of compounds based on similarities or differences. Thank you. Information. To ensure that its identified via its are valid before subsequent time and resources are invested in optimization activities, several steps are applied in the workflow. This involves retesting activities in the same assay format as the primary screen to confirm that the activity is reproducible. Concentration response assays are then employed to measure the concentration of test compound that brings about 50% of the maximal effect. A range of counter screens may be used to determine whether potential hit compounds demonstrate artifactual behavior if they possess unwanted mechanisms of action, or whether they also affect a number of related targets whose modulation should be avoided for selectivity reasons. Typically, compounds are then grouped into clusters representing the active compounds from each interesting chemical series.
Mechanistic characterization. Biophysical methods are extremely valuable in helping to carry out in-depth characterization of protein-ligand binding interactions. They often provide a simple way of compiling biochemical approaches in providing kinetic, thermodynamic, and mode of action information. For example, kinetic binding information can be obtained directly using SPR, whereas traditional enzyme kinetics experiments are required to access weight contents for slow binding interactions.
This often establishing time course under sustainable concentration of substrates, for which the control is linear, and then observing the slow decrease in enzyme rate as the test compound equilibrates with the target protein. These types of experiments are the consuming and often can be difficult to analyze to extract the relevant rate constants controlling ligand bond. A further example is describing order of addition of substitutes to the enzyme. Discussion. There are a range of biophysical methods that are available for application at many points during early drug discovery. Each method has its own advantages and disadvantages that lead to different applications, different upon the reagents available and the information content desired. For example, ASMS is most suited to higher throughput screening, whereas is positioned to deliver kinetic data. NMR can be provide structural insights, and the appeared approach for primary fragment screening, and it provides a rapid thermodynamic characterization of the binding event. The decision about which method or combination of methods to employ can be complex and subject to change depending upon particular projects, even if they are following similar hit identification and lead generation process. The choice should be ultimate focus on what information is required, the numbers of compounds required from which data needs to be generated to provide this information, and the timescale required to deliver this data. Of course, consideration of the target itself and the nature of the binding site being targeted may also influence this decision. Ultimately, practical limitations around protein availability, costs will also need to be taken into account. However, projects often benefit from the impact that a combination of biophysical and biochemical assay derived information can provide, and biophysical methods should be integral part of any protein-centric drug discovery project.
From what my teammates have talked, we can conclude that biophysical methods have evolved from being employed to address isolated issues with tool compounds or target proteins experienced in some early stage projects to now being an essential and integral part of workflow positioned to establish and pursue hit identification and characterization across the whole portfolio. Over the last 5 years or so, the progression has been driven by impact these methods have had, an increased throughput for some methods, and the recognition that a better understanding of the regrets, tools, assays, and that mechanism characterization and differentiation of hit yield a more efficient early stage drug discovery process. This has led to a more focused use of biophysics alongside to more traditional approaches such as enzyme and cell-based assays, which has increased the quality of these early fit finding assays. Biophysical methods also are increasingly used as primary hit finding approaches and no longer just for small fragment based screens but also screening increasingly large compound libraries. The recognition that compound binding can be placed ahead of activity based screening in an orthogonal dyad has been embraced by screening groups, and it may be especially useful for novel targets whose functional activities is unknown or difficult to assay. As a result, biophysical methods will remain a key facet for increasing drug discovery success. Thank you.